Hello, everyone, and welcome to the Human Cell Atlas Biological Networks kickoff meeting. This meeting is going to feature the liver and breast biological networks. So the seminar goals are to increase the biological network visibility and spark new opportunities for collaboration and engagement. It's to promote coordination with other networks, such as the CZI seed networks. And it's to lay the groundwork for drafting Atlas roadmaps in each biological area in preparation for work sessions at the virtual 2021 HCA General Meeting. This seminar will take place on the second Thursday of each month from 10:30 to 12 p.m. Eastern Standard Time. We also are very grateful to the Chan Zuckerberg Initiative, who is making this seminar series possible. So, questions. We ask that any questions you may have that you go to slido.com/HCA Bio Network, or you can scan the QR code, which you can see at the top right of the slide. So questions will be prioritized, so we encourage you to vote for questions. And you can do that by going to the Q&A tab on the Slido link. We also encourage you to participate, so I'm going to open up a poll really quickly. So we'd like to ask the audience here, what would you like to get out of this seminar? So I'm going to give folks a couple of seconds to respond to this. So I see collaboration. I also see progress in other biological networks and to reconnect. Increased knowledge of liver networks just came in. Fun facts. Collaboration and skill. Ooh. I also see a big one, overview of what is going on, which technologies are being used. Great. So the next question that we have is, how did you hear about this seminar? And this is really to help us understand how we can better reach our audience and new audience members. So just take a second to fill that out for us, please. It looks like colleague or friend is most popular right now. Great. And finally, the last question here is, are you part of any of the biological networks? So we're about 50-50 split. Oh, exactly 50-50. Okay, great. And you can refer back to these questions at any time. So going back. So the seminar format, so we will open with a talk by Alicia Jane Twigger. We will go into a 5-minute Q&A. So please do submit questions while Alicia is talking so we can address those during the Q&A. We will then have a liver talk by Sonia Mcfarland, followed by 5-minute Q&A. After the talks and Q&A, we will then go into our breakout session rooms. Those of you who did get a spot in the breakout session rooms should have received an email from meetings at Human Cell Atlas yesterday with the Zoom link and password for those breakout session rooms. Unfortunately, we are at capacity for the breakout session rooms, so if you did not sign up ahead of time, not to worry, you can still rejoin us here in the webinar for the report back session, which will take place 11:50 to 12 p.m. And with that, I want to introduce my co-organizer, Ellen Todris.
Hello, everyone. And I would like to welcome our first speaker representing Breast Bionetwork, Alicia Jane Twigger. Alicia Jane is a research associate at the Department of Pharmacology within the University of Cambridge. She began her career at the University of Western Australia, where she gained her PhD examining cells in human milk. Upon completion of her studies, she received a fellowship from the International Society for Research into Human Milk and Lactation to enable her to cross the globe to work at the Helmholtz Center in Munich. Here, she was further accepted into the Helmholtz Postdoctoral Fellowship Program, working in the Human Mammary Cell Research Group led by Dr. Christina Scheel. Subsequently, she was invited to join Dr. Walid Khaled's research group in Cambridge, where she continues her work on human mammary cells using single-cell transcriptomics. Alicia Jane, please take it away.
Thanks so much. I'm absolutely delighted to present here today. So I'll be discussing some of the one of the projects from the Human Breast Cell Atlas, one that I've been involved with particularly. But there are many chapters to the Human Breast Cell Atlas, and the one in the UK is led by my supervisor, Dr. Walid Khaled, along with John Marioni and other members of our research group, as well as collaborators with particular help from the Breast Cancer Now tissue bank. So in the healthy adult mammary gland, in its nulliparous condition, the mammary gland is non-functional. However, it does have a ductal network as well as lobes at the end of these. And during a process that occurs during pregnancy, the mammary gland is then primed for milk production. And this occurs during lactation. So following milk production, the mammary gland then returns to a somewhat resting state in a process called involution. Now, this is quite interesting because in comparison in breast cancer, the aberrant condition, there are many factors that seem to be related to what is happening for individuals in the healthy state. So for example, factors such as parity and long lactation actually decrease the risk of breast cancer, whereas other factors such as age, germline mutations, or hormone replacement therapy can actually increase the risk of breast cancer. And so we're really interested as part of the Human Breast Cell Atlas to try and understand the cell composition and how it might change due to different factors for an individual such as age and parity. So a project that's launched with the UK chapter has begun sequencing, and the data has been collected and is now undergoing analysis. But for this particular project that I'll talk to you about today, I'm focusing on the nulliparous or parous breast, along with cells in milk that we use as kind of a proxy to understand cells from the lactating breast. So just as an overview of how the mammary gland looks, as I mentioned, we have ducts, and at the end of these are lobes. And this is surrounded by specialized stroma. And if we look a little bit closer at these lobes, we have lobules. And lobules are actually made up of two major cell types. We have both the luminal cells, which become functional during lactation, and we have the basal myoepithelial cells, which are contractile around that. Now, in the past, people have separated out these different cell types using FACS analysis, using a limited number of markers. So for example, CD45 and CD31 are used to exclude the immune and endothelial cell compartment. And if we then focus on the lineage negative compartment, we can use EpCAM and CD49F to then separate out the different epithelial compartments. So we have hormone responsive luminal cells, luminal progenitor cells, as well as basal myoepithelial cells. And of particular interest, the luminal progenitor cells have been thought to be the cell of origin for many very aggressive breast cancer subtypes. So that's something to keep in mind when it comes to cataloging these cells. Now, in my lab previously, a single cell experiment to try and understand the changes of the epithelial compartment through different stages of pregnancy, lactation, virgin, as well as involution has been conducted in mice. And so we have published this previously to try and understand basically what's happening in the mouse perspective. Unfortunately, of course, we can't do the same developmental stages in adult humans. And so as part of my project, I'm instead using non-lactating breast tissue and human milk. So the non-lactating breast tissue has been obtained from women who have undergone elective aesthetic mammoplasties. The tissue is then mechanically dissociated, it undergoes enzymatic digestion, and then from there we get single nucleated cells. In contrast, from human milk, we simply just centrifuge the milk samples and undergo some washing steps, and from there we can also get out single cells. So cells can be isolated from both non-lactating breast tissue as well as from human milk. Now, if we put the cells in culture, so the non-lactating cells in 2D culture, it's been well shown that they will proliferate and form colonies. And similarly, we find the same with human milk cells. Now, if you put the cells into a floating collagen gel condition in single cells, they'll actually proliferate and branch out to become 3D mammary organoids. And we can actually do the same thing with the milk cells. So this kind of gives us an indication of the differentiation capacity of the cells, as well as show that they're viable. So live tissue and milk cells can be cultured in 2D and 3D models. This kind of brings me to the aim of the study. So clearly when we are growing the cells, it's quite interesting to look at their differentiation capacity. However, we really need to characterize the starting cells that we're starting with. So the aim of the study is to determine the cellular identity of human milk cells and which cell types they most resemble in the non-lactating mammary gland. So if we just use FACS analysis using the same scheme I showed you previously, in the non-lactating mammary tissue, we have 2 luminal populations, 1 basal population, along with the stromal compartments. This is quite different if we look at the milk cells. So we don't actually see a clear luminal population, nor even a clear basal population. So it's not quite as distinct using these markers of EpCAM and CD49F as it is for the breast tissue. So human milk cells have a different profile to cells from non-lactating tissue. And this kind of shows us that the traditional techniques of how we used to analyze these cells are not enough to really understand the cell composition of human milk. So instead, what we did is we set up a single cell experiment where we collected 4 breast tissue samples from women of different ages and parity, and as much as possible tried to match these to lactating individuals. And the lactating individuals provided milk samples where we could then isolate out the cells and compare them to the non-lactating breast tissue. So the cells were loaded onto a 10x genomics chip and then sequenced. And after filtering and quality control of the cells, we sequenced roughly 25,000 non-lactating cells and 27,000 lactating cells. Now, to give you an idea of how these cells are related to each other, I performed principal component analysis, where each of these dots is a single cell, and they've been colored by sample. So as you can see along principal component axis 1, the greatest variation between the cells is actually whether they're derived from either the breast tissue or either from the milk cells themselves. Now, if we arrange this in a U-map formation, we have found different clusters seem to be representing different cell types. And we've also done clustering algorithms on top of this as well. So overall, the greatest separation between the cells is due to the cell of origin, whether they come from breast milk or whether they come from breast tissue. Now, I used differential gene expression analysis to look at what genes were most highly expressed in each of these clusters to try and understand their cellular identity. And then, of course, looking at literature for different markers in addition to this as well. So we found one cluster in particular seemed to have a high expression of PCAM1, which encodes for CD31. This was found exclusively within the breast tissue, so not within the milk cells. And so this is representing our endothelial cells. We also found a vascular accessory population, again, exclusively found within the breast tissue, and also fibroblasts highly expressing the RNA decorin, encoding for decorin. And finally, we found a mixed population of immune cells. So these contain cells from both the breast tissue as well as from the human milk. And kind of delving deeper into this, we used a number of different markers to better understand what cell types we'd have. So we found a population of myeloid cells arising from the milk as well as from the breast tissue. And many of these had high expression of macrophage markers. We also found lymphocytic lineage cells from both the milk and the breast. And finally, we found a small B cell population that seemed to arise exclusively from the breast tissue. So non-lactating breast contains stromal cells where the immune compartment is similar to those that can be isolated from milk. Now, looking more closely at the epithelial compartment, we started out and found that actually using keratin 18, which is a pan-luminal cell marker, we found 2 milk cell clusters arising, as well as 2 luminal clusters from the breast tissue arising. Using a number of different markers, I'm just showing here for representation, we found that the breast tissue had 1 cluster that had highly expressed luminal progenitor marker ALDH1A3. We also found that genes associated with hormone responsive cluster, so estrogen receptor or prolactin receptor, were highly expressed in one of the clusters in the breast tissue. And we found for the milk cell clusters that they both highly expressed secretory genes such as alpha-lactalbumin and casein, which are the two major milk protein genes in human milk. And finally, we found one myopithelial cluster of basal cells, and this was again found exclusively within the breast tissue. So both human milk and non-lactating mammary tissue contain luminal cells, where milk contains two distinct secretory populations. But the question is, of course, is which mammary cell subtype do the milk cells most resemble? So it doesn't, as we can see from the previous plot, basically they do seem to be quite different, the luminal cells. But if we were to see which ones they're most similar to, what would they look like? So to do this, what we did is we used a set of data that was obtained previously from sorted breast tissue populations, and these were bulk RNA sequenced. And a follow-up study actually generated signatures for each of these different cell types. So luminal progenitor signature, hormone responsive, myopithelial or stromal scores. And this is just here to remind you of exactly each of the different cluster types. So unsurprisingly, we found that the hormone responsive score was highest amongst hormone responsive cells that we classified in our clustering. The stromal score was highest amongst our stromal compartment, so vascular accessory endothelial, fibroblasts, and immune cells. The myopithelial score was highest in the basal cluster. And what was really interesting was actually that the luminal progenitor score was high not only in the luminal progenitor cluster, but also within both of our milk cell clusters. So this kind of suggests that milk luminal cells most resemble luminal progenitor cells out of all the different cell types that seem to exist within the resting breast. And of course, we found this really interesting because we wanted to understand how luminal progenitor cells might potentially differentiate towards a secretory lineage. So to do this, we performed differential gene expression analysis and found thousands of genes differentially expressed. And what you can see here is that many of these genes were exclusively highly expressed either in the luminal progenitor cells from the non-lactating cells or from the luminal cells of the milk cells.
Alicia, Jane, 2 minutes.
Sure. So doing some gene ontology analysis to try and understand which pathways might be upregulated in the luminal cells, we found that pathways such as secretion, zinc transport, fatty acid metabolism, and immune response were high in our lactating luminal cells. Whereas the luminal progenitor pathways that were more highly expressed were associated with hormone association, epithelium remodeling, as well as cell adhesion. So epithelial remodeling genes and cell adhesion pathways were higher in non-lactating cells, and secretion, zinc transport, and fatty acid metabolism were higher in milk cells. So to give you an overall summary of our findings, human milk contains two secretory luminal clusters that are distinct from non-lactating tissue cells and resemble luminal progenitor cells. There is an enrichment of lipid production pathways in secretory milk cells and a downregulation of adhesion pathways. And what we find really exciting is that we think that this work provides a rich resource for constructing and exploring biosynthesis pathways associated with milk component production and transport. And in future directions, we'd find it really interesting examining luminal progenitor cell-like cells from human milk. We think it might be useful as a preclinical tool for early detection of disease. And secondly, that functional mammary genes activated in lactation could be more closely examined in metabolic diseases such as low milk supply and diabetes. Now, I'd like to acknowledge the work from both the research groups that have helped make this happen, the Schill Research Group at the Helmholtz Center, along with the Khaled Research Group at the Stem Cell Institute and University of Cambridge, along with our funding bodies and collaborators, and for you for listening. So many thanks for your attention.
Thanks a lot, Alicia Jane. Wonderful talk. Please do post your questions if you have any in the space underneath. And we already have one question. We probably only have time for a couple. So the first question is, do you think the cell type proportion identified by single cell experiment truly represents the cell type proportion of the tissue? What do you think about the inter-individual, inter-sample variation of cell type composition?
I think that's a fantastic question, and I think it is very hard to answer. I think it depends on how the tissue is dissociated, so whether each of the cells are actually fully dissociated out or not. And I think that depends on, as I said, the digestion scheme. Having said that, I think the efforts towards doing the sectioning and then the spatial, looking at the basically when they do the laser dissection of the tissue, I think that's a really fantastic way to give us an idea as to whether the proportions are similar to what we see in the transcriptomic data alone. So I think that's an excellent question, and hopefully we can talk more about that in the breakout session.
Great. Okay, here comes another. Russo has shown full pregnancies able to modify breast stem cells. He described a stem cell signature. This molecular modification was suggested as a protective carcinogenic mechanism. So, have you had the opportunity to look at the molecular modifications induced by pregnancy at single cell level?
I think that's, yeah, a fantastic question. I personally haven't done it myself, but I think it's something that hopefully the Cell Atlas in general will look at. I guess single cell ATAC-seq might help answer this along with single cell transcriptomics. But for my work personally, no, I haven't done that myself.
Okay, we have time for a couple more. One question says, sorry, I didn't get it. Did you use the single cells with or without that rebuild the 3D structure of the dissociated cells? Very nice work.
So could you repeat that one?
Yeah, and I'll also paste it for you in the chat. So, sorry, I didn't get it. Did you use the single cells with or without that rebuilt the 3D structure of the dissociated cells?
Without the 3D structure. I guess this person's probably referring to whether it was fully dissociated and whether we somehow kept a map of where the cells were coming out of the tissue. And no, we just kind of minced the tissue and then did enzymatic digestion to get out single cells. So we didn't have the 3D structure maintained in that case. It was just doing the sequencing of the single cells.
How do you know that cells from human milk truly reflect cell type composition in lactating breasts?
I think that's also another wonderful question. And of course, based on the data that I have, I can't answer that question either. But I think it'd be really interesting to go into animal models to see whether the cells that actually come out in the milk are exactly the same as the cells that are within the lactating breast. Of course, when it comes to proportions, as I mentioned, we only have immune and luminal cells coming out in the milk as well. So it's obviously only a small fraction of the cells within the lactating gland.
Does human milk contain any subset of stem cell, and how pluripotent are they?
That's a good question. I guess this is a follow-up from a few different papers. From my research, using the markers that have been previously used, I cannot see the same proportions of stem or progenitor cells. It could be that there are some plasticity of the luminal progenitor like milk cells. However, I don't see the same as what's been reported previously. And I think, in my opinion, that's because with single cell transcriptomics, obviously we can have a much bigger picture of exactly individual cells and their whole transcriptomic profile. So I think that just the sophistication of the tools has led to this conclusion.
Mm-hmm. Thank you. So we will keep the questions going, and we will keep this link alive so that Alicia Jane could answer offline. And I think with this, we're ready to move on to the second part of our seminar today. And I'm happy to introduce Dr. Sonia Mcfarland. She is our speaker for the Liver Bionetwork. Sonia is a scientist at the Ajmer Transplant Center at the Schwartz-Reisman Liver Research Center at the University Health Network, and an assistant professor at the University of Toronto's Department of Laboratory Medicine and Pathobiology and the Department of Immunology. Dr. Mcfarland's research program is focused on translating fundamental knowledge about the immune biology of the liver into clinical applications. Sonia and her research team are using advanced genomics, including single cell and single nucleus RNA sequencing, to describe the microenvironment of the healthy and diseased human liver. Her team is examining how the liver immune environment can be therapeutically targeted and manipulate it using nanoparticles to slow or reverse ongoing damage. Sonia, please take it away.
Thank you very much, and thank you to the organizers for inviting me to provide this update on the work that we're doing to transcriptionally profile the healthy liver as part of the Human Cell Atlas Liver Biological Network. So within our network and within my lab, we're very interested in understanding the building blocks of the human liver. We're interested in understanding the cells that make up the function of the human liver, including its profound metabolic function and its ability to break down drugs. In particular, my group is interested in the liver as an immune organ, and we're interested in looking at how immune cells within the liver maintain homeostasis. So the liver is a very impressive organ because of its ability to regenerate. So within the transplant program at Toronto General, patients can come in and donate up to 66% of the liver, and that grows back within 12 weeks. However, in patients that are afflicted with chronic liver diseases such as non-alcoholic fatty liver disease and viral hepatitis, this normal regenerative process is delayed or impeded. And so what we're interested in doing in the lab is looking at the functional units of the liver. And this is a liver lobule with a central venous area and a periportal area. And what we're interested in doing is understanding how cells that cycle through the liver and are recruited into the liver are interacting with parenchymal cells such as hepatocytes and endothelial and cholangiocytes that make up the bile ducts in order to maintain homeostasis. And of course, we're not alone in this effort. We're part of this wonderful CCI seed network and the liver group. And this is a very collaborative and super active group. And some of the work that I'll show you today is in collaboration with the PIs in this wonderful program. And so within our program and our group, we're interested in looking at the diversity of human liver across a lifespan. And today I'll mainly focus on the adult liver and some of our efforts to understand the normal diversity within the adult liver. So first, I'm going to start off with talking about the experimental platform we're using to understand cellular diversity in the human liver. And I'll talk about a study we published 2 years ago to generate a comprehensive cellular map of the human liver to understand the experimental platform. And this study provides a foundation for single-cell liver disease studies. And so this work is in collaboration with my team. We work on liver immunology with Ian McGilvery's team. They work on cellular biology and surgical approaches, and Gary Bader's computational biology team, all in Toronto. And so the fundamental basis of our dissociation protocol is that we try not to disrupt the hepatocytes mechanically as much as possible. So within the program, we have access to liver caudate lobes from humans that are donating their livers in the transplant procedure. So what that allows us to do is to cannulate the tissue with its own vasculature and introduce buffers, first of all, to flush away blood, and then to introduce enzymes that allow us to disrupt the cells and disrupt the tight junctions so that we can favor mechanical or enzymatic dissociation as opposed to mechanical dissociation. So this allows the cells to be disrupted, and then we open the mesenchymal layer of that piece of tissue and release the cells without very much mechanical dissociation. And so because we don't have, we are hoping to maintain the integrity of the hepatocytes, we also don't clean the cells up with columns or with magnetic bead-based cleanups. And so what we do in the analysis is that we remove dead cells by removing cells that are high in mitochondrial transcript percentage and low in the library size. And so what that gave us was a map of 5 livers from 5 healthy individuals who were donating their liver as part of the transplant procedure. And what we saw were multiple populations of hepatocytes, multiple populations of macrophages, and multiple populations of immune cells. And so this work was then followed by wonderful papers from our network, and then these studies showed resolved the fibrotic niche of the human liver, looked at the healthy margin of colon cancer metastases, studies that showed very nicely the cellular components of the fetal liver, and most recently a single cell atlas of human liver from development of disease with HCC samples and 4 human fetal liver samples. And within our team, there's multiple papers that are in the pipeline, so it's a very exciting group. So what I want to talk to you today is about our efforts and some unpublished data that we've generated in our effort to expand out our human liver map. And so we're asking the question, building on our understanding of the different cell populations that we should find within the liver, how do we understand the normal variability of the liver? So the questions are, how do demographic characteristics, age, and sex impact cell populations in the liver? And how do medical confounders such as time on life support, whether the organ is eventually rejected or not rejected, influence the transcriptomic profiles we're getting? And so this is work that's been spearheaded by a postdoc in my lab, Tallulah Andrews. And within this work, she's looking at 160,000 cells from 20 individuals, 50% female, 50% male, over a range of ages of 18 to 68, and a range of weights or of BMIs. And so within this data set, she's used multiple approaches, including Harmony dataset integration, to integrate all this data and to ask the question, how do donor traits affect cellular profiles? And so this is the human liver map 2.0. And what we see within this liver map is, again, because of the gentle handling of the tissue, you see a large proportion of hepatocytes making up this map. And as well, you can see different types of liver sinusoidal endothelial cells and different types of macrophages, as well as immune cells within that liver, or this liver map. What's interesting, as Tallulah incorporated Harmony dataset integration, what we found was that, as opposed to the first map in which certain hepatocyte clusters were very patient-specific, what she's finding in this map, or showing in this map, is that most clusters contain cells from all samples. So it allows us to annotate these populations with a bit more confidence. And what she's also able to do is take subclusters and use principal component analysis and Varimax factor analysis to look at how the impact of sex-related differences in individual populations within the liver. And so this is an example of the Varimax analysis for macrophages within the liver. And what this analysis captured was a dimension of variability that was related to male versus female differences in macrophages. So in blue, you see pathways that are high in female. In red, you see pathways that are high in males. And so signals related to innate immunity, interferon beta, are higher in females than males. And so one of the things we really want to do, because of the fact that we're seeing additional populations of hepatocytes, and those hepatocytes are conserved amongst the 20 donors, we want to look at spatial transcriptomes to understand where these different hepatocyte populations are found spatially. And so we're using the Visium platform, and this is work that's being done by Tallulah and Hca, both postdocs. And what Tallulah was able to do, and she'll talk about this in the breakout session, is again use PCA analysis to look at the dimensions of variation within this data. And what she found was the largest dimension of variation, or the highest variance explained within the spatial transcriptomics data, was the difference between central venous and periportal genes. So if you look at these little green areas, this is the central venous region. And if you look at this red area, it's more periportal. And some of these central venous or pericentral genes are CYP1A2, CYP2A1, CYP3A4, and these were also found in mouse by the Hca Events group. And as well, the periportal genes, including CYP2A7, have also been highlighted previously. So we're able to take some of these clusters and look at them, or figure out where they are spatially. But we also are encountering some challenges. So we really want to address the challenges of providing a healthy map as a comparator in liver disease. And so the biggest concern is that disease tissue can be very hard to dissociate to a single cell level because of fibrosis. So one of the things we want to address is, how do we provide a healthy comparator for samples that are fibrotic or that cannot be dissociated using single cell RNA sequencing. So in collaboration with the Aviva Gavin or at Rosen at the Broad, our team has initiated a study to look at single cell RNA sequencing versus single nucleus RNA sequencing for healthy human liver. And so in terms of the experimental the platform, we use the same liver dissociation protocol that I showed in that first video, in which we use a very gentle dissociation with the tissue's own vasculature to release those cells. But at the same time, as we cannulated that tissue, we took a small piece of tissue for snap freezing, and we dissociated them with the different detergents, or released the nuclei with the different detergents described in the Slifer et al. Paper in Nature Medicine. And so what we wanted to look at was evaluate the two different protocols with the following questions, that main questions that we asked, which were which cell populations are better captured by each technology, and how well are hepatic biological pathways captured by each technology. And so first of all, we needed to integrate this data. And so what we found was when all the samples were scaled together, first, you still saw major biological differences between the transcriptomic profiles when you compared single cell versus single nucleus RNA sequencing. And again, you did see batch effects when you compared, or technology specific batch effects. So green is single nucleus RNA sequencing and red is single cell RNA sequencing when the samples were scaled together and then the Harmony algorithm was applied. However, when each sample was individually scaled separately and Harmony was applied, these cells integrated very well together. And so what we saw was a map of integrated single cell and single nuke RNA sequencing data. And what we found was that single nucleus RNA sequencing allows for better capture of parenchymal cells. So if we looked at that original map, of the 5 livers, and even in the single cell RNA sequencing data from this data set, we saw small populations of liver sinusoidal endothelial cells and small populations of stellate cells, as well as cholangiocytes. And so within the single nucleus RNA-seq data set, we were able to capture larger populations of each of these populations, and then relatively less populations of some of the more dissociation insensitive cells. And so when we looked at the hepatocyte populations that were captured by single cell versus single nucleus RNA sequencing, first of all, we found that hepatocyte populations, major hepatocyte populations were captured by both approaches. However, when we looked on average at the transcripts, or the number of transcripts captured in using single cell RNA sequencing versus single nucleus RNA sequencing, there was about 10 times more average reads per cell for populations that were captured by single nucleus in green versus single cell in red. And then when we carried out pathway analysis, what we found, and this is the pathways that are enriched in single nuclear RNA sequencing are in red, and the pathways enriched in single cell RNA sequencing are in green, what we found was that that single nucleus RNA sequencing enriched hepatocytes with elevated metabolism pathways, including catabolism, sterol biosynthesis, and organic acid metabolism from all 3 zones of the liver. So it suggested that single nucleus is probably a better way to capture hepatocytes. And then what we saw in contrast to that is that all immune populations were better captured by single cell RNA sequencing. And this kind of makes sense from a perspective of single cell RNA sequencing skews away from the hepatocyte population. So you'll have a relative enrichment of immune cells because the dissociation techniques are more damaging to hepatocytes. So what we saw was that in terms of the single cell versus the single nucleus RNA-seq map, the maps, or the single nucleus RNA-seq map, was more comparable to what you'd expect in tissue. So many more hepatocytes versus immune cells. And then finally, what we found was that single nucleus RNA sequencing lends enough resolution to identify subpopulations of hepatic stellate cells within the liver. And if we looked at our original map, there were very few hepatic stellate cells captured. And again, these cells are likely more insensitive to dissociation. And so we were able to capture more populations of hepatic stellate cells and annotate them as quiescent activated or inactivated hepatic stellate cell.
Sonia, 2 minutes.
Yep. So with that, I'd like to thank our Chan Zuckerberg Initiative Human Liver team. It's a wonderful group, very collaborative. And then I'd like to thank my lab and all the people that have contributed to all this work, including also Dr. E. McGilvery's lab, Dr. Gary Bader's lab, the UHN transplant program, obviously the Chan Zuckerberg Initiative, the Human Cell Atlas, and the Ajmera Transplant Center, as well as all the different participants and our collaborators for all their efforts. And I'm happy to take questions.
Wonderful. Thanks for a great talk. Yes, let's move on to questions. The question that got most votes so far is the very next factor analysis was very interesting. Is this method used for the study available for others to use?
Yes, I think, and I think Tallulah will talk about the Varimax factor analysis approach during the breakout session. But this is an approach that has been developed and worked on also by Gary Bader's student, Delaram. So, and this is something that we'll chat about a bit more during the breakout session.
Great. Thank you. The next one is just like other solid internal organs, it is very difficult to get enough amount of fresh human fresh human liver tissue for single cell experiments. Also, as far as I know, hepatocytes tend to die very easily, further complicating liver single cell experiments. Do you have any tips regarding this problem?
Yeah, so this is a really good question, and it's one that we talk about a lot in that original paper. So when we first started dissociating human liver before doing single cell RNA sequencing, what we really wanted to do was to plate hepatocytes and to be able to look at them in monolayers. And what we found was the more you manipulate the cells to clean them up, to remove the dead cells, the more they die. So what we try to do in our preparation of the or the tissue homogenates is we try and favor enzymatic digestion first, and we use very little mechanical dissociation. And what we found was that when we tried to use a dead cell removal kit, for example, or we tried to use gradients to isolate different populations or to enrich for the more viable cells, we lost hepatocytes in every kind of step along that trail of cell manipulation. So one thing we're really finding is that when we use single nucleus RNA sequencing, we're getting much better hepatocyte signatures. And then for single cell, kind of our approach is, manipulate the cells as little as possible, and then use bioinformatics at the end to then remove the cells that we think are low quality and that have been damaged.
Are different levels of alcohol consumption a confounder for sex differences in expression levels for pathways and for inflammation levels?
I don't think within our data we're using alcohol levels we have reported alcohol levels for these patients. These are organs, though, that are being prepared for transplantation, so they have to reach a certain criteria. But the patient demographics or the patient information that we have is a lot of it is either reported by the family or we do have blood testing. We do test for drug levels and alcohol levels, but we haven't added that into our analysis yet.
Okay. How do you deal analytically with multinucleated hepatocytes in your single nucleus RNA sequence assays?
Yeah, so that's an excellent question, and it's one we talk about a lot. Right now, we're not able to answer that question. One of the things we're thinking about doing is trying to do isolate cells in a multi-well plate, so and identify the multinucleated cells, then disperse the nuclei and look at how the nuclei from multinucleated cells are different than mononucleated cells. But it's a really, really good question.
And probably the last one or two more. So regarding Regev lab protocols, those buffers leave behind ER pieces, and that is the key of their higher level of genes nucleus and UMI nucleus. However, how do you know all the nuclei have the carried over equivalent amounts of non-nuclear cytoplasmic pieces? Have you checked that?
Yeah, so we have tried all the different buffers, and what we found is just like the Regev lab that in TST, you see some still carried over endoplasmic reticulum. And so I think in the post or in the analysis, we're gating out those high kind of mito clusters and just focusing on the low mito cells.
How do you account for the fact that hepatocytes are multinucleate cells in your single nuke sequencing?
Yeah, so that is a really excellent question. Again, we haven't been able to do that. One of the things that we could do, or that we're thinking about doing, to look at the multinucleated cells is to use a spatial transcriptomes profile that is higher resolution. So the nanostring digital spatial profiling, because then we can get down to 10 micrometers. Right now, it's an important question, and I think it's something we should chat about during the breakout session and see if other, how other people are also addressing this.
Thank you. And please keep asking your questions, and hopefully Sonia will answer them offline. Christine, take it away.
Yes, so right now we will be going into our breakout session rooms. Just to give a brief overview, so the breakout sessions will take place now between 11:15 and 11:50 a.m. Eastern Time. You will be muted upon entry into the room, and we ask that you keep your microphones muted unless if you are a breakout session leader. Breakout session leaders will call upon you for questions and comments, so please utilize the hand raise function, or you can just raise your hand like this, and they will call upon you. Once you're finished speaking, please remute yourself. So you all should have received breakout session login information and passwords yesterday. If you did not, unfortunately, our breakout session rooms are at capacity. So for those of you who did not get a space in the breakout session room, please feel free to rejoin the webinar at 11:50 a.m. I don't want to take up too much time, so please now go to your breakout session rooms.
Okay. Hi, everyone. I'm Hca Events from UC Irvine. I'm one of the PIs on the CZI network for building a human breast cell atlas, and was one of the breakout session leaders for today's session, focusing on the breast, together with Walid. And so I think we had a nice, active discussion today. And also one thing that became clear is that this was probably one of the first meetings that we had as a group, and I think this is very helpful to get people together and to notice who else is working on similar projects in building a breast atlas. So we went through a couple of different specific questions that we posed to the group, and had a fairly informal discussion on each of these points. So one of the first very general points was what are the goals for the atlas, and what was mentioned is that really kind of similar to probably most of the other atlases, we're trying to really build an unbiased reference of cell types and states with spatial information that will ultimately allow us to go forward and really depict what changes in disease. And one of the most common diseases is obviously breast cancer, but there were also other diseases mentioned, such as lactation problems, etc. Inflammation of the breast tissue. So I think that was very clear coming forward from the group. Other things that were mentioned were really deciphering the variation between individuals. So there's a very distinct life cycle of the breast tissue, and it's a very hormonally regulated tissue. So there's a lot of metadata that becomes very important. So we need to have information as we collect these samples. We need to have information on, for example, parity status or menstrual cycle status, menopause status, etc. So really the importance for the metadata was mentioned during this discussion. And then I don't know, Wally, do you want to add anything to this?
No, I think I agree, and I think it would be good to I think redouble our efforts in terms of getting everyone involved those who are doing Atlas projects for the breast to kind of get in touch, and maybe we can put things together, and maybe start a conversation, a bit more of a regular forum, so that we can learn from each other, I guess, a bit more how we're doing things in the different labs. And eventually, I guess we can end up with a more comprehensive atlas, which is beneficial for everyone involved, for everyone out there who wants to obviously learn more about breast disease. I think the other thing that came out, which was very interesting, is to, at some point, it would be very good to have some sort of a reference point in the breast of where the samples are collected. And I think the Nick's suggestion to maybe understand how plastic surgeons or cosmetic surgeons actually decide on that, so maybe tap into the way that they decide on things, and the maps that they use might be a starting point for the atlas. And I think that's probably kind of information that's not being collected at the moment by most tissue banks, for example. But I think definitely the key message takeaway is that we need to reach out maybe to other groups that are doing this and build a more comprehensive group to connect.
Yeah, and I think adding on to that, one thing that was nice today, I think, is that maybe by accident, but some people from actually other groups ended up in this discussion. For example, there was Martin from the Lung Network, and he had some sort of very helpful reflections from their efforts to sort of congregate the whole group in a larger effort. And so I think it's another example where talking to people from other networks is very helpful to organize your efforts a little bit better. So I think going forward, like Walid just said, I think the key take-home message from today's breakout session is that we need to try to organize our group efforts a little bit tighter. And I think this session here today helped us getting a few names on the map, so that we can actually put all the efforts together and start working on the roadmap towards building this atlas. So I think all in all, it was a helpful first step in this direction.
Thank you, Kai and Walid. Appreciate it. So let's now go over to Tallulah and Martine from the Liver Network. Please go ahead and share your report back.
Yeah, I thought maybe Alan wanted to summarize, or I think we discussed some of the spatial things. I think Tallulah, that I'll let you discuss. We also discussed some site-seek techniques and how you can get protein data from the data sets. Yeah, I think it's showing that there's a lot of questions from people and a lot of interaction. So yeah, we agree with the previous report. Yeah, it's also nice to see that multiple people are trying really different types of technologies. So will be very nice to compare at the end and to see.
Yeah, our session really highlighted how many different technologies are being developed and used throughout the network, and hopefully put people in contact with other people who, if they want to set up or use that technology themselves. I think there was at least 3 different spatial technologies as well as site-seek discussed in our session, as well as various challenges for different types of samples and different regions of interest, but how to actually handle those samples and whether particular technologies will work for that type of sample or whether you need to modify it, for instance, to deal with fat lipid droplets or to handle hepatocytes gently enough.
I think what was also key from the talk of Tallulah was and Sonia was actually the difference between cellular isolation using enzymes and Nuke-seq and so on. And it's true that if you then realize that for the site-seq, you actually need to isolate them, yeah, it does tell you that you'll have a big bias in these type of data sets. That I think would apply to any organ, so that was very cool to see.
Yeah, I hear all of that.
Alan, I don't know if you want to add the last couple of things for the liver, or maybe we leave it at this.
But I think that's that, and we will make this available as a recording for people who had to leave earlier. Please do tell them if they ask you, and I'll let Christine take it from here.
Great. Thank you, Ellen, and thank you, everyone, for joining us today for the kickoff meeting to the Human Cell Atlas Biological Networks. I hope that you learned a lot. I know that I did. We also want to give a special shout out and thank you to the Chan Zuckerberg Initiative for their generous support. We would not be able to run this seminar series without their support, so thank you. I also want to thank the HCA Biological Network coordinators and members and the HCA Executive Office, so Ellen Todris, who helped coordinate this seminar series along with myself and other members of the HCA Executive Office. We also want to thank Brian, who helped run the back end and the technical and video production aspects of this seminar. A very big thank you to our speakers, Alicia Jane Twigger and Sonia Mcfarland, and also to our breakout session leaders, Kai Kessenbrock, Walid Khalid, Tallulah Andro, and Martin Guillem. The next Biological Network seminar will feature the adipose and lung networks. That seminar will take place on Thursday, February 11th, 10:30 to 12 p.m. Eastern Standard Time. We will be notifying the community of registration on our HCA Slack channels and on our website, and also through email. So please check out our sites that way and check your email. So the HCA has 18 biological networks that are active. If you are not part of them, or if you would like to join up a network that you're not currently part of, we encourage you to do so. There's a feedback survey. If you a QR code that will bring you to that feedback survey, and you can indicate there your interest in joining up. So as Ellen mentioned, today's talks will be made available on our YouTube and Bilibili sites. So please check there. It usually takes about 2 to 5 business days for those to be posted. If you are not a member of the HCA Project Registry, we encourage you to join the Project Registry. Check the Zoom chat box on how to do that. And for more information on this seminar series, check out our website. So how can we do better? Do you have any ideas, any suggestions? We really do want to know. So please take our feedback survey. And you can do that by going to slido.com/HCA BioNetwork or by scanning the QR code. And just to end, some upcoming HCA events. So we have the HCA Developmental Computational Modeling Seminar, the Americas, Europe, and Africa edition, that will take place January 28th, 10 to 11:30 a.m. Eastern Standard Time. Check our website for how to register. The HCA Developmental Computational Modeling Asia and Australia Seminar will take place February 3rd, 7 to 8:30 a.m. GMT. And as I had mentioned, our next Biological Network seminar will feature the adipose and lung networks, and that will take place February 11th. And save the dates for these 2 really exciting meetings. They will be virtual. The general meeting, the HCA general meeting, will take place June 7th through 9th. So please go ahead and block your calendar out for that. We'll have more information coming soon. And then also the HCA Developmental and Pediatric Cell Atlas virtual meeting that will take place August 25th through the 27th. So if you're interested in attending those, block your calendars. And as always, HCA events are free and open to all. So please encourage other communities to register and sign up. And with that, Ellen, any final words? Great. So thank you all for joining, and have a wonderful rest of your day.